Sir Arnaud Marchand, are you ready? You have the floor now.
Thank you. So thank you, Joris. It's great to meet the broad COVID-19 research community. So I will be discussing current knowledge about the immunology of this infection. And there has been a lot of research and a lot of data that published over the last months, really rapid progress in our understanding of this infection, yet there are still many unknowns. And what I will do is to use some of the illustrations of some of the great reviews that have been published over the last few weeks, summarizing our current knowledge. Most of what has been studied so far is around the pathogenesis of COVID-19 and especially the role of the inflammatory response. I'll start describing our current knowledge and understanding of this and introducing the therapeutic interventions that are targeting the immune system in order to control the disease itself. What we are starting to study is what happens next. What is the immunity induced by SARS-CoV-2 infection, to answer the key question of whether patients who have been infected will be protected against a reinfection. And as a transition to the presentation by Dr. Saville, I will say a few words about the immune targets for vaccine development. So there is now clear evidence that COVID-19 as a disease has a strong inflammatory component as compared to asymptomatic patients or mild symptomatic patients. Severe patients with severe or critical symptoms have signs of heavy, strong inflammation, both at the level of the organs. Here in the lung of a post-mortem examination, we have massive infiltration of the tissues with immune cells. We will discuss those cells in a minute. So that's at the level of the organ, but also at the level of the systemic, the body itself, the body itself, where in the blood we can see that severe disease is associated with the production of large amounts of cytokines, chemokines produced by immune cells. Unusual for a viral infection, C-reactive protein at high levels, but also evidence of intravascular coagulation, where fibrin degradation products are at high level in the blood of those patients with severe disease. So why is that happening? So when we think about a healthy immune response to a virus like SARS-CoV-2, we conceive that those epithelial cells that are infected with the virus and that are undergoing apoptosis are rapidly recognized by macrophages that rapidly destroy them. And that the information that's generated by the presence of these viral proteins is instructing the adaptive immune system, leads to the production of antibodies. Among these antibodies, some will be neutralizing, will neutralize the infection of the epithelial cells by the virus. These antibodies will also help the macrophage function in a specific way, destroying the virus, and will have T lymphocytes, whether they are CD4 cells producing antiviral cytokines or CD8 T cells killing infected cells. Altogether, these actors contribute to a healthy immune response and the elimination of the virus. That's not what we see in severe diseased patients. In those patients in the organs, what we see is the accumulation of macrophages that are derived from circulating monocytes. They are recruited in the organs. We have large numbers of T lymphocytes, and those immune cells produce large amounts of cytokines that trigger massive inflammation that is disruptive and toxic for the tissue. We also have evidence from SARS-CoV-1 infection that antibodies could also play a role causing antibody-dependent disease enhancement by activating cells, innate immune cells, and promoting inflammatory response as well. So why some patients do develop severe disease, we don't know yet, but we think that what happens at the beginning of the infection, really during the first times of the infection, may be critical. And at that time, when you have any viral infection, an immune response that's really critical is the production of type 1 interferons. So interferons are very potent antiviral molecules that are produced when cells are infected with a virus. This is a complicated slide, but scheme, but I will go through it, simplifying. So when the virus is infected, the nucleic acid of this virus, in this case the RNA, is recognized as dangerous by the cell. And this triggers a cascade of events leading to the production of these interferons. They are produced, and what they do is to enable the bystander cells to protect themselves from the virus. They become very resistant to the virus by producing a number of other molecules that have antiviral functions. What we've learned from SARS-CoV-1 and MERS infections is that those viruses interfere with the type 1 interferon response, preventing the induction of type 1 interferon production and also the effect of the type 1 interferon. So what we think is that between patients who develop either mild or severe infections, a key difference could be the magnitude and the kinetics of this response, where a prompt and efficient response would lead to viral control. A delayed response in some individuals, maybe more than others, with potentially inhibitory factors by the virus itself. These delayed responses would lead to viral replication, it would allow the virus to replicate. But then when there is a lot of virus on board, there seems to be then at that time the production of large amounts of interferon, which then exacerbate the response and provoke immunopathology. So delayed and exacerbated type 1 interferon response could be key to pathogenesis. And within this further event of immunopathology, I think it's quite clear that macrophages and macrophage activation plays a central role. We do see, this is again a complicated slide, but I'll go through this slowly with you. So within the organs of severely diseased patients, what we see is accumulation of macrophages that are coming from peripheral blood monocytes. What happens is that epithelial cells, which are infected by the virus, produce chemokines, which attract the monocytes at the site of infection. If you want, they say, please come, because here locally, our local macrophages cannot do the job. There is plenty of virus here. You should come and help us. And that's what the monocytes do. They get there, they differentiate into macrophages. But this is not, a peaceful meadow that they meet. This is a full battlefield where there are plenty of stimuli for these macrophages that are arriving. The virus itself, which will stimulate them, interferons, which will then be produced in large amounts. Also, other immune cells like NK cells or activated T cells producing cytokines that will further activate those macrophages. I mentioned non-neutralizing antibodies as immune complexes that can also trigger macrophage activation. So basically, these macrophages find themselves in an environment where they receive massive numbers of potent stimuli. And their response to that is to contribute to the process, because they start producing themselves large amounts of cytokines, which will further trigger inflammation. Another component that may be critical for the role of macrophages is the stimulation of coagulation. Within the tissues, within the blood vessels, capillaries of patients with severe disease, we see thrombi and we see necrosis. So intravascular coagulation, that's toxic for the organs. What we know is that macrophages themselves can promote coagulation. When they're in blood vessels, they communicate with endothelial cells. They produce cytokines that activate these endothelial cells, which produce, like macrophages do, produce tissue factor, which is a procoagulant molecule. They attract neutrophils, which also stimulates clotting and intravascular coagulation. So macrophage activation and their interplay with endothelial cells could be really critical at the level of the organs, causing necrosis and their dysfunction. Another player that we believe could be critical in the pathogenesis of severe disease are T lymphocytes. And so T lymphocytes, under normal circumstances, would control the virus by producing antiviral cytokines, by being cytotoxic. And in order to prevent these cells to become toxic and excessive in their response, regulatory T cells will produce regulatory cytokines like IL-10. What we see in severe disease is lymphopenia. That could be related to recruitment to the organs, but we believe is also induced by their depletion. their activation-induced cell death. So what we also see is that associated with that activation, we have exhaustion of T lymphocytes. They start expressing inhibitory receptors to prevent activation, further inflammation. But what results, another result of that is that they lose their capacity to produce antiviral cytokines. CD4 T cells seem to also start producing, instead of antiviral cytokines, pro-inflammatory cytokines that further contribute to the inflammation. And also regulatory T cells apparently stop producing IL-10 or produce lower levels of IL-10. So you have fewer lymphocytes with decreased ability to control the virus, promoting inflammation, and less able to be regulated. So you can see that within this disease, cells like macrophages that are meant to do good start playing a toxic role. And this is the basis for some of the interventions that are being tested and essentially focused on inflammatory cytokines, neutralizing their activity and reducing inflammation. Something that is also critical is a more focused approach. So this approach will be non-pathogen specific, really inflammation at large, but the use of convalescent plasma containing high titers of neutralizing antibodies, and perhaps more specifically, monoclonal antibodies or polyclonal antibodies that have been selected for their specificity and their neutralizing capacity have the potential to control the virus and thereby decrease the development of inflammatory responses. So this is where we are with the disease itself and its pathogenesis. Now it's time, as Dr. Elina and Dr. Fontenay as well have alluded to, it's now a time to understand what will be the consequence of this infection. Will the patients who have been infected by the virus be protected to reinfection? So we believe that antibodies play a central role in the rapid control of viruses in immune patients, in immune subjects. So what we know, as Dr. Elina described, we know that antibodies are induced when the viral RNA is decreased. We see within 1 or 2 weeks, we see the accumulation of IgG with neutralizing potential. What is intriguing is that in patients with severe disease, we see more rapid production and the production of higher levels of antibodies, suggesting that they could play a pathogenic role. What happens next? What happens next is are these antibodies sustained? Do we develop memory B cells, which may help us respond to the virus when we meet it again? We don't know. SARS-CoV-1 data suggests that B lymphocyte memory may be short-lived. So we have to really watch this and follow these patients to see how long-lasting could immunity be. With SARS-CoV-1, we see that T cell memory is much more long-lived. And so maybe a shorter duration of B lymphocyte memory could be compensated by T cell memory. But really a huge gap in our understanding of natural immunity, natural infection-induced immunity, is really the quality and the duration of immunity that would prevent reinfection is unknown. And I think importantly, what could prevent carriage is unknown as well. We do see severely infected patients with prolonged carriage. We also starting to see patients with mild or virtually asymptomatic infection excreting the virus for a prolonged period of time. So what are the immune determinants of carriage control will be very important to determine. So what are the targets for vaccines against SARS-CoV-2? Main target is the induction of neutralizing antibodies, obviously high avidity antibodies direct against the receptor binding domain. That's for sure the vaccine should do. There could be a potential interest of having, in addition to neutralizing antibodies, the induction of cytotoxic T lymphocytes. As I was saying at the beginning, if antibodies neutralize the virus primarily outside the cells, cytotoxic T lymphocytes are helpful in rapidly destroying the infected cells and preventing further spread of the virus. As Dr. Saville will describe, we have a number of different vaccines that are being developed with traditional formulations, with live attenuated or inactivated vaccines, recombinant subunit vaccines, with adjuvants, so those are the traditional, but also more recently developed, more novel, innovative ways of immunization, and these include the recombinant viral vectors and new nucleic acids, whether DNA or also, importantly, RNA. And if all these formulations are meant to induce antibodies and are likely to induce antibodies, neutralizing antibodies, they are not equivalent in their capacity to induce cytotoxic T cells cytotoxic T cells with either the live attenuated or the recombinant and nucleic acid-based vaccines with a higher potential to induce these cytotoxic CD8 T cell response. I've been asked to briefly mention the possibility that pathogen-agnostic immunity could be important. This is based on some epidemiological studies suggesting that BCG is not a vaccine against COVID, but a vaccine against tuberculosis may help the host preventing severe infection. And this would be based, we don't know how this would work exactly, but this could be based on what we call trained immunity. And that's training monocytes, macrophages, innate cells, to better fight the virus when at the beginning of the infection. So there is a potential for training the immune system in order to prevent, in a non-specific way, the development of severe disease. And there are, as you may know, several trials that have been initiated, randomizing exposed people to BCG or no BCG. So what could be the take-home messages of current knowledge of immunology of SARS-CoV-2 infection? I think there is strong evidence for a role of immunopathology in severe disease. As in other infectious diseases, we have a dual role for a number of these players, whether macrophages, T lymphocytes, or antibodies. They are meant to do good, but something happens that we yet need to understand that drive them to become destructive and to cause immunopathology. The immune determinants of severe disease are currently unknown. Risk factors, major risk factors have been mentioned: age, sex. Sometimes young people develop severe disease. What could be the immune determinants of severe disease in these patients is unknown and will be the subject of research for the future. We have identified targets for therapeutics. As we understand the disease better, it is clear that new targets will be identified and that more refined and perhaps more specific strategies will be developed. Humoral immunity is a key thing, or immunity at large is a key thing. Will people be protected from reinfection or carriage is central, and we will have to watch this very closely. And I think the roadmap for vaccine development is relatively clear. We need neutralizing antibodies. There may be a role for cytotoxic T cells. We don't want disease enhancement. I mentioned the role of antibodies in promoting a disease. Maybe an appropriate T cell response may do the same. So we'll have to watch this carefully. And I think that if the vaccine could prevent not only infection, but also carriage, that would be an advantage for prevention of transmission within the population. So thank you for your attention.
Thank you very much, Professor Marchand.